just illustrates how important biology is. But why else are we interested in spatial biology? There's at least two additional features that I'm really excited about and that made me focus at my lab focus on spatial biology. It's not only the location. One, I think it has really tremendous multiplexing potential where from minimal tissue amount you can go to a maximal output. And I just want to highlight one thing we did very early on, where we have two core needle biopsies. These are tiny core needle biopsies from breast cancer patients. It took us, I think, over a year to optimize a single cell dissociation step from that whole core needle biopsy. So in the past, actually, you need a lot more tissue. We were like super excited, like, hey, we can make like one beautiful single cell RNA-seq data set from a tiny core needle biopsy. Having said that, this was about 5,000 cells, looked great, but all the tissue was poof, gone. That was it. We cannot do anything, we cannot validate anything. Luckily, we have two research core needle biopsies. So for that core needle biopsy, we decided to try something else. We said, like, let's use that for spatial transcriptomics. And we generated a spatial transcriptomic data set from that one. We generated an IMC data set from that one. In addition, we have H&E, we did spatial attack-seq, all from the same core needle biopsy, same size, except here we got 5,000 cells. Here, theoretically, we get 1 million cells. Like the whole biopsy can be represented, not just at the transcriptomic level, but you can look at the epigenomic level. So that just illustrates like how powerful this, spatial omics technologies are for multiplexing and maximizing usage of tissue, which is especially important in a clinical setting where you cannot just ask for, hey, can we get another research biopsy? They're all going to say no, hell no. And then the last part, which is somewhat associated with this one, and I will talk about this at the end of a half time, is that I think there's a massive clinical potential where whatever we do can almost directly translate it to pathology. And I think we're almost ready for digital molecular pathology to be used in the clinic. All right, so what is spatial biology and oncology? Well, it's really, like I said before, it's kind of like combines two really well-established fields. One is the imaging that we've been using for like decades, where in this case, this is a core needle biopsy. It's stained by H&E. You can see where all the different cell types. This is not cell types, where the pathologist will look at and say, okay, I see this and this is probably that type of subtype. Obviously, each individual cell has a lot more information that cannot be encoded by a simple RGB, like red, green, blue image. It has DNA, RNA, protein that together defines individual phenotypes. So with spatial omics, we can extract that, profile that, and put it back in its original location, giving us the opportunity to go one step or multiple steps beyond that and basically identify what are the individual cell types, what are they being expressed. There's a B cell, there's a T cell, is it an activated T cell, is it a regulatory T cell, whatever you can think of. And as I said before, why is it so impactful? I think you get a lot more tissue, you get a lot more information per tissue, as I described previously. It can work with any sample of tissue nowadays. It doesn't matter whether it's super tiny, it doesn't really matter how it's processed these days. We have come a long way working with FPE samples, and we can generate really large data sets really fast. I think in the past, people were excited when, hey, we have 1 million single cells. In a single run, with enough multiplexing, enough samples, we can probably generate a million cells, or information about 1 million cells in a single run. That's a single day. All right, having said that, so my lab is, I think, a very multidisciplinary lab where we focus on spatial biology, but we really tackle it from all different directions, so we're trying to stay ahead of the curve. Where a lot of our questions are around oncology, system biology, technology implementation, new technology implementation, think about how to combine them, but then also the open source software to analyze this type of data, because it is very complex data, and we need to think about how can we analyze the data, how can we make it as simple as possible for anyone using it. This is a team right now that is doing all the amazing work, a really talented and very driven team that has done a lot of like piloting of different technologies, different projects. And so today I want to talk about 3 stories. I just want to make sure that there's enough time. And feel free to interrupt me, ask questions during my talk. I want to talk about how our lab develops a software to create your own, what we call a Google map for cancer research. So you create all of that data. It's a massive amount of data, but how do you use that? How do you use that to derive new insights? I want to talk about our osteosarcoma metastasis paper, or that's coming up, and I want to end with something that we're really excited about, spatial transcriptomics, and bring it to the clinic, or bring it potentially to the clinic in breast cancer research, breast cancer disparity research. All right, let's start with this one. So this one is somewhat a continuation of the work that I started as a postdoc, where I had the pleasure and the fortune of working with Long Kai, who is one of the grandfathers of spatial transcriptomics. He developed seqFISH Plus. And so we had to, that was basically a spatial omics technology where from each individual cell you can measure hundreds, if actually not thousands, of individual RNA transcripts. And we had to come up with a way to analyze that data. So I developed Giotto, which is an R package. And that's being used now all over the world, has been cited over 900 times right now. But it was actually really focused on, we said, hey, this is like single cell data with spatial coordinates, which was a very big oversimplification, which we realized when I started my lab and I started discussing with my first genetics and genomics program student, George. And we thought about, hey, we need to extend this idea, and we really need to think about what is spatial biology, how can we represent it, so we can make it future-proof and give it to everybody. And so we came up with an idea that was supported by Chan Zuckerberg Initiative, and it's a foundation program, and they only support what they consider essential open source software. So we were one of the lucky that got supported by them. So we're going to go through this paper, but this paper was very recently published in Nature Methods, and he, together with Jocelyn from GC Wen's group at Mount Sinai, are the co-first authors on this paper. So what is that? What do we do in Giotto? So in Giotto, the Giotto suite, in this paper, we really thought about, okay, so how do we need to represent spatial data, or spatial omics data, and make it so that you can actually use it with any other or upcoming technology, and even combine different spatial omics technologies. The first thing we realized is spatial biology compared to single cell is not happening at the single cell, it's happening at multiple levels. You can go all the way down to individual transcripts where they're organized in organelles, and the single cell is part of a larger niche or whatever it is, and that's part of a larger tissue structure, right? And then we thought about, hey, there's different technologies. They all have their advantages and disadvantages. Some work at the higher or lower resolution spatial arrays. There's multiplex, there's mass spec, there's multiplex imaging, and there's like just typical sequencing based. We want to be able to represent all of them. So we had to come up with like new, and all of this is software development, but that's kind of like what we need if we work on the front of spatial omics biology. We had to come up with ideas to how do we represent all these different data types, including images, points, intensities, shapes. We had to think about what are the tools we need to integrate and create this, what we call this vertically integrated connectome, where you can integrate all these different layers, including transcriptomics, epigenomics, metabolomics, if you have that. And is that data really necessary? This is kind of like outdated because there's a lot more papers. I think yes, there's a lot of papers coming out right now, and I already illustrated this in the beginning. Since you have your tissue, you can easily generate different spatial modalities from each individual section and have that kind of like being collapsed, where you generate your multimodal data and you understand your tissue at multiple levels, at the protein level, at the transcript level. And these are just a couple of these papers where they're trying to do this. This, for example, where they merged electron microscopy with spatial transcriptomics, which was pretty cool. And they have some other ones where we're also starting to work in 3D and having spatial attack-seq being part of the story. So what we came up with, and apologies if you're not really a software biology expert, but this is kind of like some of the things that we did is we decided let's separate the features that you analyze. The features are your transcripts, your proteins, and the spatial unit, because a lot of these technologies happen at different units. Sometimes it's like a 55 micron spot, sometimes it's an individual transcript, sometimes it's an area of 10 micron. And so we had to find out a flexible way to combine all of that. And so if we do all of that, then we have this kind of like framework where you can combine any spatial technology with any other spatial technology. It doesn't really matter about the resolution, it doesn't matter about the feature. You can kind of like vertically integrate that, and you can represent it as a whole, which then gives you the framework to do any type of like downstream algorithm development. Not going to go into these details. This is just kind of like the blueprint of some of these package development that we do, and where do we store the data, and how is the data, how do we need to keep track of the data. There's a lot more that goes into something like this than just like, oh, let's do this. Well, I would say right now with Gemini and ChatGPT and all its little friends, you can probably do this, except at some point the whole thing will collapse because you need to have kind of like strong fundaments. Importantly, I think a lot of these technologies, they're coming out with their own tools, and it only works with their own data sets. And we, from the beginning, were like, this needs to be technology agnostic. It needs to work with any type of technology, and every type of technology needs to be able to be represented and to be able to merge. And so we just show that with different technologies, like there's spatial DBT, spatial genomics. These are some of these like smaller technologies like SlideSeq, none of the 10x. It can work. So it can basically work with any technology. So I'm going to give you some key examples of what we can do, and after this we're going to go a little bit more into biology, so hold on. If you're not interested in this part, there's going to be other stuff for you. I'm not going to go through everything, but it's a little bit long, but just to show what our framework allows you to do. For example, as I said to you before, if you have a spatial transcriptomic data set, there's actually multiple scales you want to look at. Don't just limit yourself to a single cell. That's basically what most people do, because like, hey, this is a single cell with coordinates. There's a lot more you can learn. You can, if you have Murfish data or Xenium data or anything where you have spatial imaging based methods, so you have information about individual transcripts and then you have some kind of like cell segmentation information. If you have enough information, you can identify nuclei, you can identify cells, but you can also use algorithms, including in Shoto, but also other algorithms, to, for example, identify similar niche regions and spatial domains. So all of these are somewhat related to each other, right? And we can represent them. We can identify where the nuclei, where the cells, where the spatial domains, and how are they kind of like correctly organized, and how is that related to each other. And then a very simple comparison you can do is, let's say you have that kind of like level of understanding that you can visualize. Maybe you're interested to understand like what is the difference between cell and nuclei. And the reason I'm giving this as an example is because we're all interchangeably using single cell RNA-seq and single nuclei RNA-seq as one of the same thing. We're like, yeah, it doesn't really matter what we do, it's the same thing. I think this little example shows you that you need to be a little bit careful about this assumption. And so what we can do is we can identify the nuclei, we can identify the cytoplasm, aggregate the transcripts that are associated with these two different cellular structures, and see what is enriched in one versus the other. And one thing we see is that there is very clear enrichment of different transcripts in the nuclei, where it's a lot more about, in this case, this is in the cytoplasm, it's a lot more about luminal, endoplasmic reticulum, that sort of thing, while in the nucleus, it's like cell death, regulation of cell death, which makes sense in this specific data set because this is a data set from breast cancer patients. So this is probably some apoptosis, active apoptosis happening within these cancer cells. What you could also do, just to basically illustrate what I was saying before, let's say we take the whole cell and we only take the nuclei and we only aggregate the transcript in the nuclei versus the whole cell. So now we're actually comparing in the same experiment, which you cannot do, by the way, if you have your own single cell and single nuclei. We compare single cell information with single nuclei information. How does that correlate to each other? So you can generate that kind of like data, and then we can basically see for the cells, the whole cell and the nuclei, is there a one-to-one relationship? I think you can clearly see that if you just clustered it unbiasedly, there's not a one-to-one relationship. And I think it's very important because one, and this is conceptually, a whole cell gives you understanding about kind of like the steady state of that cell, because there's a lot of transcripts happening in the cytoplasm that are there for maybe a few days even, while the nuclei gives you information about what is being actively transcribed. And so that's why it is, not surprisingly, that's two separate things. But it's also important to understand that, because then you know exactly like, in which direction are they going. And this is a little bit of trajectory inference, velocity type of analysis. Like in which direction is your cell going versus what is the steady state of your cell. Another example, and this is basically what I was saying before, and things that we do in our own lab, is that what if you have multiple layers of information? You might have your favorite immunofluorescence antibody staining that might be adjacent to your Xenium or Visium HD or Visium panel, and you want to integrate those two. You want to understand, like, around these foci, for example, that I found, what is being enriched, what is being expressed. So the first thing you need to do, and it's not really easy, is you need to align these different data sets. And we took this data set from Tenix themselves, where they have single nuclei, they have Visium, and they have Xenium, and they have IF data for some key markers. And we built a tool that allows you to align all these data sets so that you could directly start comparing your Xenium, your Visium, and your IF data in a vertical manner. And by the way, this is the George that I introduced before. He is the main author, but really almost everyone in my lab had a big contribution to this paper. And this specific part where we built a Shiny app to make that as easy as possible for people to align their data was driven and developed by Jungjin Zhu, who is another genetics and genomics student. And so basically you have to define these landmarks, and then you can kind of like do this automatically. You can align your data so that your Visium and your Xenium is nicely aligned, and you can do some type of downstream analysis. In this example, I just want to see how much time I have. I just want to show one thing you might be interested in is if you have IF data, and we have IF data of CD20, which is a marker for B cells, and HER2, which is a very specific marker they use in breast cancer to identify if you have HER2 positive cells, for example. And then you have your gene expression that you got from your Xenium data, and you just want to know, like, hey, are they the same? Are they spatially associated with each other, which is something you would expect. You don't necessarily, it doesn't necessarily have to be true. So you can do that, and then, for example, one thing you see here is depending on the bin area that you use, that information is quite variable. And understanding that principle is pretty important to understand, like, how are two features, protein and RNA, spatially associated, and how does it change with the area that you look at. Another thing you can do, if you have that Xenium and Visium data, which I think some of us have, because Xenium, and I didn't really go into details, Xenium is super highly resolved, very sensitive, but limited to 500 genes that you select. Now you can actually go to 5,000 genes, but it comes with sensitivity loss. While Visium and Visium HD now is, to some extent, unbiased. It takes your whole transcriptome, but doesn't have the same sensitivity. So they both have their advantages and disadvantages, and depending on what you want to do, you can do either one, or you could say, hey, let's do both, and we have the best of both worlds. But one question we had is, like, is there any bias in the genes that you profile with Xenium versus Visium HD? And we looked at, do these genes correlate with each other? Do they spatially correlate? And there are certain genes, for example, it's FASN, very high, nicely correlation, where we can make a pseudo Visium data set from Xenium. That's something our tool can do. And we can directly compare that with the Visium. But then there's other genes where we noticed, hey, Xenium is pretty good at picking it up, but Visium is pretty bad. And we were wondering, is this intrinsic to the technologies, which we initially were like, yeah, that's probably a problem with the probes, or there's a problem with the polyanylation of this transcript. It turns out the majority of the correlations, the poor correlation, which you see here, this is just correlation scores. The poor correlations really nicely are explained by just average expression levels. And so all of that, what you see here is probably HTC is very lowly expressed. The Xenium is just very sensitive because it's in situ hybridization based, while you just get that kind of like the sampling bias from the Visium approach, which is polyadenylated. You need to kind of be able to capture that. So I think a lot of what people say, like, oh, a lot of it can just simply be explained by expression levels. Again, why, and this is also the reason why I like Xenium so much, is it's such high sensitivity, so that depending on whatever transcript you're interested in, that can be a very lowly expressed transcription factor, it will most likely identify and find it. Right. Another thing that a lot of people have struggled, and some of you here might have struggled with the same, if you have a spatial data set, you need to, and especially this is again spatial imaging based Xenium, you need to be able to segment your cells, which means you need to be able to identify the boundaries in order for you to know which transcript belong to which cell. That is part of these technologies. The good part is that there's many tools to do it. The bad part is they're all giving you different results. And so here on top, I have 1, 2, 3, 4, 5 different tools giving you all slightly different segmentation results. And so these, is it magenta or cyan, whatever, these are these individual cells, and then you assign your transcripts. And then you can basically use our framework again to compare, does it actually lead to significant changes? Because that's what you're interested in. If it all ends up with the same type of like final conclusions, then there's not a big problem. So one thing we did is we used the original segmentation provided by Tenix himself, this is the Xenium data set, and we compared it to Baser, which is another approach that has been developed some time ago. And so what I'm showing you here is that we do a co-embedding, which means we're analyzing both approaches at the same time. So we can rule out any other like covariates that might be at play. And we wanted to see, does it lead to changes in identified cell types? Because that's usually the first step you do. And what we noticed is that there is, at least for 2 cell types, there's a very big difference. Like there's a 50% difference type of. And for example, it's for stromal cells and CD8 positive T cells. I think both these cell types are really key right now in breast cancer research. And again, this is a breast cancer sample. Stromal cells believed to be a major role in supporting your cancer cells, and a lot of people are trying to see, can we basically attack them, or can we treat them, or can we target them? While CD8 positive T cells, this is kind of like your main cell type, or one of the key cell types for any type of like immune response to breast cancer. So having that being misclassified by over 50%, depending on how you look at it, I think is a major issue, and at least understanding what are the limitations of your segmentation might be super important. All right, then scalability, as I said before, yeah.
Ruben, can I ask you a quick question? Have you found an example where a gene is better detected with Visium that might be missed in the Xenium platform if a probe is missing from Xenium, or is it the case where the Xenium probes that they selected just covers everything and there's no point using Visium?
I cannot say this for sure. So in general, Xenium always is more sensitive. There might be cases where it's the opposite. Now we're going a little bit in depth. There is a little bit, some people have shown that Xenium has potentially a different problem where these probes might detect off-target effects, which is also related to in situ hybridization.
Okay.
That is not something we look at right now, and so that might be something that is a Xenium-specific problem and not so much a Visium problem, at least if you use a 3' end. All right, so single-cell RNA-seq data is big. Spatial data is way bigger. And if you use the raw data, it's actually massive. It can be massive. And so one of the things we have spent a lot of effort is how can we implement different software tools, or maybe to make this easier. And one of them is kind of using a database, style processing, whatsoever. And I just want to show, this is StereoSeq. StereoSeq is a little bit like Visium HD if you're familiar. So it's unbiased. It uses, you identify fully adenylated transcripts. So you kind of like aggregate everything you can find. But it can be massive. And so this is a whole mouse embryo that they profiled in their FLAG paper. And that data can be terabytes big, so it's very hard to process that. But one of the things we did is we have a very flexible binning approach, so you can start binning at a very high level, so it's not that high resolution, so it's smaller. And you can go to a lower level whenever it's needed. So at a high level, you would see something like this. You would see overall big structure. You might be interested in this area in the brain. You can say, hey, let's just take that out, use a smaller binning strategy, and all of a sudden you can start seeing this very nicely layered brain structures. And that gives you this very interactive way of analyzing data that even is way too big for your own local machine. You can use any of our tools or other tools that are compatible with Shopto to do even like further like smoothing of the data, and you really nicely see this like multi-layered brain cortex region that you expect to see. As I said before, our tool is so flexible that you can make a pseudo-Visium if you want, and that allows you to do one thing. If you create a pseudo-Visium from that data, you can use that as the ground truth to test or benchmark deconvolution algorithms. So deconvolution algorithms are these tools that if you don't have single cell data, but you have single cell information, then you can use that single cell information to try to deconvolve, for example, a Visium spot into multiple fractions of these individual cell types. This type of approach gives you the opportunity to test multiple of these deconvolution approaches and just see which one agrees with the ground truth, which is your stereo-seq dataset or Visium HD dataset if you have that. All right, now I just want to have a very small pivot. So we have this pretty complex way of doing all this kind of big data analysis. So Eddie is another genetic genomics student. So hey, can we just make this a lot simpler and don't have to deal with all this like, hey, let's style, let's do variable binning. Can we just come up with an idea that it just works just on your local machine, not on a fancy supercomputer, but on a local machine? And the reason why that's necessary is with all the data that we're currently generating, and this is just Visium HD, stereo-seq, it's all way beyond the memory limits on your most personal computers. So you have to kind of like move into a supercomputing environment or a large server in order to analyze the data, which I think leads to what we sometimes call research disparities, that only people that have access to that and have enough funding can do it. Well, everybody should be able to do it in a local machine. And so we came up with an idea, and that's using these latest online analytical processing databases. So where your data is not necessarily anymore, doesn't necessarily have to be in RAM, in memory anymore. You can actually live on disk. Your disk can be a lot bigger. That's easy, like a terabyte nowadays. But maintaining the same levels of speed, and that is important. That is what these new embedded databases allow you to do. It's some kind of like SQL databases, but not just for retrieving data, it's for online analytical processing of data. And so Eddie started working on top of that and actually built different classes, including for doing anything on geometrical level data, so spatial geometries, any type of points, polygons, whatever. Also matrices, but also in genomic sequences, and that's DB spatial, DB matrix, DB sequence. These are different packages that can be your low-level backend for any type of like large-scale spatial data analysis. The DB sequence gives us another additional advantage, something that not a lot of people do, and that we started exploring, and that's especially relevant now with the latest Visium HD 3' that is available, where it's Visium HD, so it's very highly resolved. It's 2x2 micron, that's its highest resolution. But it gives you information about 3', so it uses 3' polyadenylated transcripts, so it's completely unbiased. What we don't use, we use it just simply to identify how many genes do I count. But there's also a stretch of 3' information at the end that we don't use, and that is your 3' end of your gene, where polyadenylation happens. So now we can also start looking at, do we see changes in polyadenylation? And a lot of people kind of like don't even think about these ideas, but a lot of genes have multiple polyadenylation sites. So the polyadenylation sites are the sites where your gene decides, like, hey, I'm not gonna be further transcribed, that's it. There's a lot of binding sites for microRNAs, for RNA binding proteins. So depending if you have a shorter or longer, it might have a big effect on, for example, transcript stability. That might have an effect on protein stability. So there's a lot of information that is encoded in this 3' end that we can now start to use. This is something that we're actively working on. We hope to get out pretty soon. But what we, for example, can start doing is not just looking at the gene counts and identifying cells, but also identify cells that potentially have different polyadenylated transcripts, and are they potentially related to cancer aggressiveness, RNA binding proteins. So this is an active work by Eddie. All right. We have a lot of interoperability. We interoperate between the bioconductor spatial experiment, between Seurat, between SquidPy and the Python community, spatial data, another Python community. We are built on top of, the reason we like R, by the way, is R has a really big and long-standing spatial data science ecosystem, which has been around for decades. Like spatial biology is kind of like new, but we're just reinventing the wheel oftentimes, because like spatial ecology, spatial geology has been around for decades, and they are very far advanced in terms of like spatial statistics. And a lot of times what ends up happening is that we're re-implementing some of these ideas. So by building on top of these previous packages, I think we can see a lot further. And just to give you an idea, this is something that I think all different spatial analysis that have been built into, I think this is Terra, which is one of these tools. And just to give you an example how fast you can build something by using their interpolation techniques. So interpolation is an idea where you can infer missing information by basically have 2 points of information, and you think there is some kind of like continuity, and so you can interpolate what's happening in between these 2 points. It's being used in the weather industry, where you have these weather ration stations, and you want to identify what's happening in between. So just to give you an idea, this is a Visium dataset, an old Visium dataset. These are these spots, 55 micron. We identify one specific gene, PANTR1, and we want to say, hey, can we do this at single cell level rather than the Visium spots? And so we can identify the nuclei because there is a matching H&E. We can convert the gene expression, this PANTR1, to a raster. We can interpolate, that's what I just discussed. So you go from something like this to something like that. Now we can use our tool and our framework to overlay the nuclei information, or expanded nuclei information, with the interpolated gene expression data. And so all of a sudden you go from something that looks like this, your Visium, your typical like spots, to something that where you have now, it's not exact, but it's a proxy at the single cell level. You have that type of information. So if you have your Visium data, for example, you do that for all genes, you go through a whole pipeline, you go to something that looks like this to something that looks like this. So now even you can, with established tools, you can go from your original Visium spot at 55 micron to individual cell type level information, which I think is pretty impressive. All right, with that, I want to conclude this. So there's Joplin Suite. There is this website if you want to learn more. We have a lot of tutorials. We have a lot of additional workshops that you can attend. If there's a bug, let us know. Let us also know if you're happy or if you're not happy. All right, now I want to shift gears. How much time do I have?
15 minutes.
Oh, 15 minutes. All right, this probably doesn't take too long. So this was another project, and actually started by Jason, another genetics engineering student in my lab, who's now full-time in Sarah Mazzulli's lab, and now being taken over by Veronica and Jeffrey. And the whole idea was here, like, we want to look at osteosarcoma. So osteosarcoma patients with osteosarcoma have a big problem once there is metastatic lesions into the lungs. And so what we see is that there's a very low overall survival in patients, and oftentimes very young patients with metastatic disease, where the survival really drops really low. And it's even more precarious if you notice that after 1980s, there was an increase in survival because of all the introduction of chemotherapy and kind of like multiple chemotherapy at once. But then in the last 3 decades, that survival has stagnated. Nothing really benefits these patients. So if these patients, the moment they have metastatic disease, it's almost always fatal, or oftentimes it's fatal. And this is where we got these ALSF grants called the Crazy 8 grant. Is, I guess, one of the biggest foundation grants for pediatric cancer research. And we are interested in understanding why osteosarcoma, which is a primary cancer of the bone, why it kind of like goes to the lung. Not necessarily why it goes to the lung, but how can we understand what happens in the lung? Because the idea is that it goes to the lung really early in this micrometastasis, but then why does it develop, and why is it so refractory? Why doesn't it respond to chemo at all, or to many new type of drugs that we're trying to develop? And the tools we're using this really simple mouse model where you can inject F420 cells. F420 cells are mouse osteosarcoma cells. We inject it in the tail, they develop metastasis, and we use our Mersco platform to analyze the type of data. I just want to say 2 things. F420 cells, they are very homogeneous. There's a little bit of like subtyping potentially, which is mostly related to the number of detected transcripts, but overall, pretty homogeneous cell line. And then the readout we do is MERFISH. MERFISH, like Xenium, is this kind of like spatial transcriptomics-based methods where you identify individual transcripts, and then there's a cell, but you can look at the bigger cell, and you can look at the whole tissue. So that's the kind of data. All right, so what do we detect? When we are able to generate data, it looks really beautiful. It's a little bit hard to generate Verscope data, I need to be completely honest. It took a long time to generate it. Xenium, a lot easier. But when it works, it's really beautiful. And we're able to identify all major cell types. We identify all major cell type part of the lung structures. Think about the AT1, like the alveolar type 1, alveolar type 2, but also like the endothelial cells, everything that you need to see. But you also identify these predominantly yellow, these are the metastatic sites. The first thing we had to figure out is where are all the individual lesions? And this is very similar to how patients represent themselves. There's many individual lesions, and that identification of these lesions is actually not trivial, because you can actually see, you don't really know exactly where does one lesion start versus the other one. And so we had to develop some kind of like supervised way where first we had to identify spatially organized processes and identify cell types that are spatially organized. And so once we knew these spatially organized processes, that we could start tracing individual lesions, which was also like a very kind of like repetitive, not repetitive, but very like we had to go through multiple iterations. And I think Veronica actually did a lot of work there trying to identify these lesions. And now we had to do some additional refinement at the end because you have some edge problems. We did some refinement with k-nearest clustering, and that these are all the individual lesions. So now we have identified these lesions, and our first question was, are these lesions different? Are they the same or just at different stages, depending on size or whatever it is? And so what we wanted to do is we wanted to further subtype the individual cell types in order to understand are there compositional differences. So the first thing we did is we integrated our MERFISH data in red with a big single cell RNA-seq data set from a similar tissue here in blue. We did harmony integration and we did label transferring, which again is all part of the Giotto suite framework. We did that also at individual cell levels to further subtype, and this is, for example, macrophages, where our approach is, and this is something that is not trivial, is we first do it on the MERFISH data only. We do the label transfer, but the label transfer is a little bit dirty because the single cell and the spatial data is not a nice one-to-one match. So we have some kind of like manual readjustment of clustering results based on that label transfer. And at the end, we do a majority voting, like what is the most likely cluster? And so we end up with what we believe is probably the best informed clustering result, matching both the MERFISH data and your single cell RNA-seq data set. So we can use it kind of like interchangeably. So with all that information, I just want to show if we highlight some of these major cell types, like the tumor cells, the macrophage, epithelial, activated endothelial, we do see already very specific or lesion-specific biology happening. For example, if we look at number 6 here, we see that there's a lot of tune expression, which is not seen in any other lesion. So that is for the cancer cells. If we look at other cell types, for example, we look at DATPs, which are these damage-associated transient progenitors. So it's a state in between these AT1 and AT2 cells, major cell types of the lung environment. It's sometimes identified in some lesions, but not another one. Sometimes excluded, or sometimes it's included. We don't know exactly why this is happening right now. Same with macrophages. There are certain macrophages, interferon gamma macrophages, that are part of some lesions, but they're not part of other lesions. And then we also have endothelial, activated endothelial. Again, some lesions do express them, and it's associated with tumor angiogenesis, while others don't. So this is already indicating that, hey, there is a lot of like lesion-specific biology happening. Then we wanted to see, is there also intra-lesion differences? Do you have your lesion, but is that lesion kind of like homogeneous, or is that different by itself? And for that one, we focus on these lesions, and we consider spatial co-expressed modules. So you can look at individual genes, they have a certain spatial co-expression profile within these lesions, but we can identify these modules. And these modules are genes that show similar spatial co-expression events in all of these lesions. And so we can use that, and when we do that, we actually see 3 main modules plus a few smaller ones that are associated with the core, with the edge, and the periphery. And so there's also some biology happening, intra-lesion differences within these lesions that are associated. You can nicely see here where the colors are the core, the edge, and the periphery. So we see structural events within the lesion, and then we also see differences between lesions. And then the next question we had, okay, let's combine both information and see, can we learn something about, is there really, what is happening at the biology level? And so here we clustered all of these individual lesions by combining the compositional differences between lesions, but also combining the spatial differences, like are these cells that we identify, are they in the core, the periphery, or the edge? And one thing we noticed is that if you just put them all in a human map, well, they don't really nicely cluster together. There is some clustering you can force, but it just showed that it takes up the whole space. There's a lot of like gene expression that kind of like, is it a stochastic event or not? We don't really know, but it takes up a whole gene expression space. It doesn't seem to be very strongly related to size of, because that's something we hypothesized. Maybe it's just a size, maybe it's just a stage of that specific lesion. It doesn't seem to be the case. Another thing we considered is the proximal, or the proximity, the physical proximity between 2 lesions, and the similarity that we have here, this tree, similarity based on compositional differences that I said before, is that associated? In other words, are 2 lesions that are found close to each other, are they typically showing the same biology, or is it more independent? If it's independent, that means that every lesion has some kind of like a free will and can decide to do what it is. So what we did is we did a simple test that if it's perfectly spatial means that it's all explained by spatial proximity, so just everything that is close by, it should look like this. And we combine this close, medium, far. That's kind of distribution you expect. If it's completely random, then that is what you expect to see. That's completely randomized, where the close, medium, and far relationships are kind of like all over the place. There is no strict separation. And this is our real data, where the real data seems to suggest that it's actually mostly random. And again, I think all of that together, I think we're showing, I think one of the first spatial evidences that there's different biology happening at all these individual lesions, and it's all happening like originating from the same cancer cells. So it's not necessarily intrinsically driven, because I already showed you at the beginning, the cells that we inject are pretty homogeneous. And so the tumor microenvironment really seems to play a role in orchestrating some type of like biology, and then is there some stochasticity happening? We don't really know. What we do believe is that this underlies some of this treatment resistance that patients see, because all of these lesions can have their own environment, and some of them can be receptive to a treatment while others don't, and there's always some lesions that are growing. And this is where we are right now with that. All right, now maybe I'm gonna rush to the last one in a few minutes. It is pretty cool, but it's pretty small.
May I ask you?
Yes.
So do you think different cancers have different heterogeneity? So this one, for example, hard to treat because it has many potential things to survive, and others that will be more homogeneous in this.
I don't really know.
Yeah.
The one thing that I didn't went over with osteosarcoma is special in the way that it has a lot of chromosomal rearrangements, where this is kind of this chromotripsis happening, and there's also some other that is not specific to many other cancer cells, which I believe at least contributes to some of the cancer-specific plasticity that it has. Whether that cancer-specific plasticity is also associated with the lesion biology happening, I'm not sure. And that also goes back to some of the other cancers that I'm not familiar with. Do they have the same issue or not?
But it will be developing in that direction, maybe, if somebody's interested, or how you see this.
Yeah. Well, right now, I think what we would hope to do is, can we do something similar, but with other type of real, this is a mouse osteosarcoma, can we use human osteosarcoma? Problem is that if you inject that in mouse, you need to use you don't have the full immune system, so you're a little bit limited to that extent. Right. So we also do a lot of breast cancer disparity research. And my lab, together with Naomi Ko, who left to NYU, we established a whole new clinical breast cancer program going all the way from patient consenting sample collection and doing anything that you can think about with breast cancer research. We recruited a lot of patients over the year, but collecting primary tissue is a really difficult process. And so Jeff, who was a postdoc in my lab, he was talking. We were thinking about, how can we potentially improve this? Because how do we get primary tissue for research? We get these procedures, but then only very few patients are being approached. Then are they being consented, and do we collect? And the other problem is, from the available research, tissue research, if we look at the genetic ancestry, is that it's predominantly white, while there's underrepresentation for underreported or underrepresented patient populations. So some of the barriers for getting that primary tissue for research. So this is a research-specific biopsy. Some of the work I showed you before. This is not the diagnostic clinical biopsies because they need them all the time, especially in breast cancer, but also other types. So there's logistical issues, very difficult, especially with BMC, for example, which is not primarily focused on research. It's like clinic first. There's patient hesitancy. They don't want to give additional research for multiple reasons, oftentimes really good reasons. Oftentimes there's also a tissue availability issue. When you have a small tumor and you have a couple of 1, 2, 3, sometimes 4 research biopsy for clinical use, there's just nothing left for research biopsy, so there's just no way. So we looked at the pathology pipeline, and one thing we noticed is that during pathology, they actually throw away some sections because they want to go to a next level and they don't really care and they just throw it away. And we went through them and it took some time, but it's like, hey, can you not throw away, but can you instead give it to us and put it on one of our slides so we can do spatial transcriptomics with that in-between section. And so these are diagnostic biopsies, right? So they're always there. We generated that one, and that if it works, it would give us the option to do method development, really think about, hey, how can we establish molecular digital pathology? We can start building atlases because we could potentially do this with every single patient, and it could be the beginning of real personalized medicine because we have information for every patient and we can give you the ideal treatment. All right, so here is a typical breast cancer core needle biopsy in the H&E stain, and that is from an actual patient here at BMC that was being used by the pathologist to identify what kind of subtype, breast cancer subtype this is. We said, hey, don't throw away these adjacent sections. Can you put them on this section? And this is just the example. And then we can identify all the different cell types in the same way as we did before. So this is again a UMAP. We can identify all major cell types. So we didn't change anything, by the way. We just followed whatever they did. We didn't use any RNAs free, whatever. We just give it to us, don't change anything. But that's oftentimes a problem with pathology. There are very specific standard operating procedures, and this whole thing took exactly 1 minute for one of the histotechnologists. And after that, we just took over and we did everything ourselves. And so now we can put this back, and this is an adjacent section, not immediately adjacent, but very close. You can immediately see the similarities, and while they can see beautiful purple and pink, we can see exact cell types. We can see exactly where the cancer cells, what are the T cells, what are the B cells, everything you would like to know. And then obviously the first question is, does it recapitulate the actual pathology report? And for breast cancer, they look at very specific markers, for example, estrogen receptor, progesterone receptor, Ki67, that sort of thing. This is the IHC, the actual data from pathology, and this is our data, spatial transcriptomic data. And as you can see, visually it matches, and also just looking at the numbers, the pathologist, if they would use our data, would come to the exact same conclusion as they did on their own data. The really cool part is obviously, at least for us, is that now there's a lot more you can do. And so, for example, we can look at the spatial co-expression modules again, the thing that I showed you before, like which genes are spatially co-expressed, which means there is some spatial thing happening. And one thing, for example, one example is the interferon. There is an interferon, if you can zoom in, there's a couple of cells expressing a lot of interferon response. that is something you can, by no means, you can identify that on just pathology type of stains. We can identify which cells seem to be overrepresented and most likely contributing to that interferon response. And no surprise, really, it's the T cells. And then the question is this useful? I think it is useful. There is a really recent paper, for example, where we say, hey, this is the importance of type 1 interferon in orchestrating the cytotoxic T cell response. So immediately we can use that data. We can recapitulate what pathology said, but we can immediately go to the science and basically what is known and what can we use in the future to help that patient. What the pathology is potentially more interested in is that, and I didn't really mention that, this could replace all other existing clinical gene expression assays. There's, for example, in this breast cancer space, there's Oncotype DX, which is just 21 genes. There's MammaPrint 70 genes, ProSigma 50 genes. All of that could be in one single spatial transcriptomic panel, and we could have all of that information immediately from the start for a fraction of the cost, depending on how you organize. So I think this is really exciting, and I believe this is just showing that we are ready to kind of go into this next round of digital molecular pathology. All right, now just very quickly, I think this is my lab, wonderful lab. We did all of this. I only talked about, I think, 25% of everything we have done, and there's a lot of other big data sets, including in breast cancer, including in pancreatic neuroendocrine tumor that we're working on. These are the collaborators. There's GC's lab for Giotto work, Brian Roberts helping us with the osteosarcoma work, Naomi with the breast cancer work pathology. And there's other ongoing work. I didn't talk about some of the really cool work from Chungsheng with Dries on the neuroendocrine tumors. And we're doing a couple of other smaller projects as well, and then all the funding. And with that, I just want to say, if you want more spatial information, if you're interested in Giotto, contribute, add your tool, go to this one. There's a spatial biology core here by Sarah Mazzilli. So if you want to know more information about what is available here on campus and what they can help you with, reach out to Sarah. If you want to have a broader view about what people are doing, there is also Gestalt, which is just a WhatsApp group, but they have a lot of people helping you if you have specific problems on the computational side, on the technical side, that you could be part of. And that's it. Thank you.
Questions? I'll start off with one. Were there some images you showed with the immunohistochemistry staining not quite lining up with the spatial expression? Or like some spots where there was staining in the HC but no gene expression measurement?
Yeah, so these were not immediately adjacent sections. So that's one okay. Problem. It's probably 50 to 100 to maybe 150 microns. So we're talking about, so you can generally see that the overall architecture is the same, but we can definitely not say anything about one-to-one mapping. So right now, we just don't really know, for example, is there a limitation to our transcript identification or is it something else? But overall, the main conclusions remain the same.
And then overall, the turnaround time for this to be applicable to a clinical situation, is it still weeks to get the data? Or?
No. I mean, obviously, I think if you optimize everything, I think you can turn this around in a week. And that is more or less what an oncologist expects. You need to have your panel ready, you need to have the machine ready, and it needs to be available at all times. But I do think you can turn this around in 1 week, and that is basically everything you want. Because also the data analysis, that can be all automated, and we're actually working on something, and this paper, the last one, will go on bioRxiv really soon, where we generate all the data and then we use some AI tools to basically generate a pathology report. So that will all be automatically generated. So that's all in 1 week. We go from here's the tissue, here's the full automated report. Do whatever you want with it.
Wow.
Yes.
Typical stuff. Particularly the last bit. Have you had the chance to compare the Oncotype DX data from the same patients as your spatial transcriptome?
Yeah, you should talk to the person behind you, so that Michelle is actually doing that.
Oh.
It's probably our biggest data set that I'm not showing you, where we have done this systematically, where we have Oncotype, we have the Oncotype from patients, then we have Oncotype from 18 patients, we have Xenium data. We also generated our own Oncotype, from the same tissue. Now we're comparing, like, what do you lose by just using Oncotype, which is a very simple bulk qPCR, versus what if you have spatial xenium information? We know exactly, and we see some pretty big changes, for example, related to tertiary lymphoid structures, abundance of cell types. All of that is really important. That is a really big paper that I'm not showing today. I will need another hour. We're working on it, and we hope to have it really soon.
Yeah, I was interested in if there will be a way to look at dynamics of treatment, right? How if there is a possibility if somebody start neoadjuvant treatment, there will be biopsy before and after you can look at the tissue.
Yeah, I think yes. The issue is always, and hopefully this might solve some of it, is can you get these longitudinal biopsies at multiple points, for example, after treatment, before treatment, maybe metastatic size if you want to compare primary. Right now, that's just really, really hard to get these research biopsies.
Yeah.
If pathology decides to, hey, let's make this easier, hopefully in the future, we can just use whatever they discard. And we just need like one 10 or 5 micron thick section. That's the minimum we need. Ideally, we want more, but that's enough. And the technology is that well established right now that the failure rate is actually really, really low. At least not if you use Murphy.
Clinical trials maybe for this, where people enroll and know that they will have biopsies.
Yeah, that's the goal.
Do you think this will completely replace single cell? What is stopping that from happening?
Well, maybe. I think not yet. I think there is a role for single cell. So oftentimes we actually combine single cell with per spatial. So spatial, there's some issues with spatial. It all depends how you look at this. Xenium, for example, you give Xenium very high resolution, but you only have 500 genes. If you go to 5,000 genes, and we have a comparison between the 500 gene panel and the 5,000 genes, your sensitivity drops by 5 to 10-fold. So you don't have that problem with single cell. So with single cell, you have your single cell. And so integrating the two is right now giving us the best results. Now, if you have all the money in the world and you generate not just one 10 micron section, but you generate 10 or so, so you capture the whole cell, because we're only capturing one small section, but you want to capture everything, you're going to get pretty close to what single cell can offer you. But that is at a very big cost. So right now, combining the two is probably the best solution and what I recommend to most people.
Last question?
What?
Yeah, you can.
So, Xenium gives you 5,000 genes. How practical it is to expect that you take these sets and match it with existing databases, single cell analysis, to kind of reconstruct the whole cell types and impute or whatever the remaining genes that are not on the panel?
Everything comes at a cost, but you don't need 5,000 genes to identify all cell types. You don't need that many genes or markers to identify cell types. But then it goes back to, you need to have ideally matching single cell or single nuclei RNA-seq data, and then you can impute it. And that, I think, works really well. It's obviously an approximation, because you don't know exactly if this is the same cell or just an adjacent cell. But I think we're getting pretty good at that. And we're getting pretty good at, and this is something, for example, we also do in this, I didn't discuss this, in the osteosarcoma, also in Michel's work, where we know more or less what are the most similar single cells to my spatial cell, and you can use that to do gene cell enrichment analysis. So again, you use both data sets and to the best capabilities of what they can offer you.
Okay. Thank you, Ruben, for a great seminar.
All right, thanks.
And for the students that are having lunch with Dr. Dries, they'll be meeting up in K3